Welcome to my world. Another day, another time, another period for knowledge acquisition. Knowledge is power, health is wealth. Today we shall be talking about HIV because we are in the season. Every December 1st is a reminder of HIV/AIDS, the journey so far. So I titled this HIV, how far? How far have we gone? HIV still remains very, very relevant. It refuses to grow. At times you want to wonder why it is so. The prevention is very clear, but man's indulgence in all these things that satisfies the flesh is another issue entirely. But regardless, I am trying to make effort at letting us know the progress that has been made in the medical science field, the medical laboratory science effort at getting rid of these menace, HIV/AIDS. Journey with me as we go into the slide section where I shall reach you more with the updates on HIV. The outline. We shall be talking on the objective, the introduction, historical fact at its eradication, lab diagnosis and prevention, especially, and conclusion to end it all. The objective. To expose the medical laboratory scientists, medical fraternities, WHO's, UNESCO, PEFLA, AIDS, UN AIDS, etc., their effort at curtailing the HIV/AIDS menace and the possibility of its eradication by 2030. The introduction. HIV was isolated in 1983 by virologists from the Institut Pasteur in France, and ever since, efforts were made to convince people that what it caused is AIDS, and AIDS was real and deadly. The mortality rate as a result of doubt and stigmatization was high, and stigmatization is still the cause of the new incidences of HIV that are available because people won't want to go for tests to confirm, and that allows them to spread the infection. And it spread worldwide and became a pandemic. At least 10,911 people died of HIV in the US in 1988. In 1995, the death rate from HIV per 100,000 people was 16.2. It slowly declined, increasing from 2.6 deaths per 100,000 in 2010 to 1.3 deaths per 100,000 in 2021. If we continue stigmatization and people are not going for counseling and testing, these achievements would not reduce any further. The target of eradicating HIV/AIDS by 2030 will now remain a mirage. The decline in rate of death is a concerted effort from the medical fraternity, research institute, medical laboratory scientists, international organization, USAID, WHO, CDC, UNESCO, PEFAB, the acronym for United States of America President's Emergency Plan for AIDS Relief, etc. HIV is not an easy virus to defeat. More than 600,000 people still die every year from the virus because they don't know they have HIV and are not on treatment, or they start treatment too late. This is despite extensive adoption of WHO treat all model and treatment guideline, which I shall expose some part of as we go on later. Yeah, this diagrammatic expression starts the historical facts on the effort made at its eradication. When you look, every part of this virus is labeled. It does not have a cytoplasm nor a nucleus. All are not defined, but it survived because it's able to make use of all these component units. HIV involves all its component parts labeled above to perfectly replicate itself for survival. The two glycoprotein receptor GP120 and 41 are used for attachment and penetration of host cells. Once they are able to penetrate any cell, then they are able to move in and remove the glycoprotein that covers the naked virus genome, that RNA. The RNA will now be able to replicate itself because it has a reverse transcriptase, the RNA reverse transcriptase that will now convert and use the DNA of the host to do that work. Yes, when we look at the historical perspective, in 1994, a study showed that providing antiretroviral pregnant women infected by HIV a short course of treatment for the baby as well, once born, reduced transmission to below 5% from a height of 15 to 45% without treatment. So the PEPFAR, that is the Presidential Emergency Plan for AIDS Relief, and the VCT started immediately. It was like an order that people should now be voluntarily counseled and tested, so that the pregnant women especially and their spouse in some countries like Nigeria were now the target. They were tested, and by that way the population of the percentage of the incident gradually reduced, and the prevalence also. The availability and coverage of antiretroviral to prevent HIV transmission from mother to children has been remarkable. So that really affected 80% of pregnant women with HIV able to have access to antiretroviral. By 2015, WHO recommended treat all, where anybody, both those who are gay and transgender, that they are already people suspected to have HIV, were just given the antiretroviral drug to prevent HIV acquisition, and it is called pre-exposure prophylaxis. WHO also announced 3x5 initiative with a target by 2005. That is, in 2020, their target was, 2005, the 3 million people would have been covered with the antiretroviral drug. 2014, they introduced the 1990, where 90% of people were tested and treated, and so it's all 1990. And they were able to achieve by 2022, approximately 86% of people living with HIV knew their HIV status, 76% were receiving HIV treatment, and 71% were really suppressed. And these targets have been extended to 2025. Yes, laboratory diagnosis. WHO wouldn't have been able to achieve our targets but for the cooperation of the medical laboratory scientists who were at the forefront of testing to confront the HIV status of volunteers all over the world, monitoring so many variables that are indicative of efficacious treatment, and declaring the viral load as undetectable where treatment is successful in suppressing the virus. The sample collected remains the patient's blood or other body fluids that may be required to perform innumerable tests listed below. Yes, number one of it is accurate diagnosis. Before you can arrive at that, you must have all your standard operating procedures, so that the result has the control, it has the standards and the test itself that you will compare to each other. So the operating standard must be very high to get accurate results. 2, viral load monitoring. The samples are collected, the appropriate serum is tested to know what the viral load is. The CD4 count is also analyzed. The CD4 count shows that once it is reduced, you have a situation where the patient cannot really combat the virus. So because the CD4 is the chemokine usually used, and then you now have a greater study now showing that there's this CCRF, the receptor 5, and the heterozygous delta 32, that they values too, which helps to really tell you that this patient is very susceptible to infection even when you cannot detect any virus in that patient. These are tests that are ongoing. Early detection and prevention are also done to confirm if the patient has HIV. You also want to talk of research and development that has helped to spread the rate of detection and also reduce the problem. Education and awareness, yes, like what I'm doing right now. Quality assurance and control of RTD kits. These are kits that has to be standardized to find out how sensitive and their specificity. Without all that being done by the medical laboratory scientists, the result you are getting, even testing yourself, is rather rubbish. Data collection and analysis so that you can know whether there is an improvement. All the percentage I've been mentioning are from what was collected and analyzed. Supporting HIV/AIDS program, that is all I am doing, and all medical laboratory scientists. Advocacy and community engagement. Yes, this is part of what it means to talk to people, to be more faithful, to abstain or reduce their, change their lifestyle and sort of the 11 to etc. You can just think of endless things, so many innumerable things that the medical laboratory scientists are involving in making sure that you live a very healthy and safe life. Yeah, prevention. In the early 80s and 90s, the prevention acronym was ABCD. A stands for abstaining from sex. Be faithful to your wife, your husband. The use of condom, though objected to by some faiths. D, like my faith. And D, don't share needles. These days, we talk about, when you talk of don't share needles, we are talking of the gay people and those who are involved in cocaine and hard drugs, so that the needle can be contaminated and you transfer this infection. These days, WHO is talking about 5-pillar PMTC, that is prevention of mother-to-child transmission, condom, exposure prophylaxis, which I had explained, essential triple. This is when you now include syphilis and hepatitis B in the test performed. That is, you include these organisms, the antigen that will detect syphilis and hepatitis B virus, and voluntary medical male circumcision, which was part of a research that found out that those who have not been circumcised could easily be infected and also transfer this infection to people who are not infected. So the recommended. In conclusion, the stage at which we are is that nobody has been totally cured and not needing drug anymore. You have heard of so many people saying the virus has been removed from them, and no, it is true to a certain extent, but they must continue their drug because this virus is very latent, like the latent TB that will hide and it will come out when the person becomes more seriously immunocompromised. The greatest challenge to curing HIV permanently is the virus' ability to hide and remain dormant in pockets of healthy immune cells, specifically CD4 and T cells. This latent reservoir can reactivate if treatment is stopped. The use of gene therapy on human stem cells, that is when you talk of doing transplanting all over, trying to produce new blood for these people, may provide long-term protection against HIV infection and progress. To be safe, stay safe. Stay by adhering to the ABC of HIV prevention strategy. If you like my presentation, like I've always said, please press the subscribe button. Subscribe, press the like button, click onto both, and you will surely be notified when the next production is out. And Thanks for listening.